living in a simulation. I won't give away any spoilers, so if you want to know, you can look at the stream that should be available on YouTube. Now, before we begin, I would like to remind people of the Twitter account at Pint of Science with the hashtag Pint20. So please, if you're interested, if you have a question, if you're enjoying it, let us know and help share the wonderful science that is undertaken already right across the UK. So, this edition of Pint of Science is brought to you by the Natural Resources Institute with the title Insects and the Lynx Effect. And this is based around the idea of communication, but not necessarily using Twitter or blogging. We're talking today about how important chemicals are in the lives of insects and how they interact with their environment. This is central to the Natural Resources Institute's mission to try to seek social, environmental, and economic solutions to challenges faced by food systems like issues associated with food security, particularly those involving farmers in the developed world. Many agricultural systems are entirely dependent on the services provided by insects that occupy an essential ecological niche as either friends or foes to our farmers, and therefore play a really vital role in controlling or enhancing the livelihoods of farmers. Therefore, understanding how insects communicate, especially using chemicals, is only going to become more important in the future as we see to try to develop innovative and new ways to adapt to climate change and improve the resilience of agricultural systems. So, before I introduce my first speaker, I would just like to ask you to submit a question in the box inside, and after both our talks, you will have the opportunity to grill, not literally, both our presenters. Okay, so our first presenter is Dr. Steven Harte and is a chemical ecologist. Before coming to the University of Greenwich, Steve, like my good self, was working in the University of Manchester, although unlike me, he was working on something really interesting, like isolating drug compounds from fungi. Over to you, Steve.
Well, thank you for that kind introduction, Connor. And yes, this is my presentation on Insects and the Lynx Effect, my chemical romance. This particular romance started around 3 years ago when I joined the NRI in the chemical ecology department, where I've worked mostly with semiochemicals. So what is a semiochemical? Well, to give it its kind of boring dictionary definition, it's a chemical that conveys a signal from one organism to another so as to modify the behavior of the recipient organism. But what does that actually mean in real terms? Well, we can split it into two. There are allochemicals, which are interspecies. So if we have a little diagram here, we have a cute caterpillar, and it might find something to feed on by the odor it gives off. But this can also be flower volatiles, and that can attract pollinators, so it can be beneficial to the plant. And then have the other side, which is pheromones, which is intraspecies, i.e. between the same species. And this is what I'm mainly going to focus on. So there are quite a lot of different types of pheromone. You have aggregation, which is just pulling lots of the same species together. You have an alarm pheromone, which if there's a natural predator in the area, they could emit. You have ovipositional, which is to do with egg laying. Territory, you can kind of see this in a lot of mammals and stuff. You may see big cats and so mark their territory. You have trail pheromones, so the most famous one is ants finding the nest. And you have the sex pheromone, which, as probably the title of the talk implies, is the one I'm going to talk about most. So how does this work? Well, possibly more or less how you would expect. So we have an example here, which is a cute little moth. And it emits a pheromone, which is a volatile organic compound. And it just emits it in the air, and it gets blown through wind. And then you have the male moth, which senses this organic compound with these antennae over here, and then attempts it to track down where the female moth is, hopefully for the male, before anyone else does. And we can kind of see this in action in this little video here, which this is apple leaf midge. And you can see these little guys going crazy because we have a pheromone in the center of this rubber septum. And just to see a trail pheromone, this is on coffee stem borer. We've actually smeared the pheromone down the center of this filter paper. And he will actually go back and forth down this filter paper, desperately searching for his way back into the nest. Spoiler alert, he doesn't find it. But moving on, sometimes there are other components that are non-aromas. And we can see that here with these cute little weevils. And they're essentially paying absolutely no attention to our lures and are much more interested in this twig that we've got in here, which this probably isn't the best example because these things are difficult to see, but it kind of indicates that even if you have the pheromone, it doesn't always work. But how do you even identify a pheromone or recognize one when you see it? Well, I'm going to go through a mini case study here of some of the work that we did at the NRI. And this was on the canola flower midge, which you can see a blow up of here, but the actual size of these midges is tiny. But they were found in Canada in these canola flower fields, which is a type of rapeseed that they grow out there. And initially, they weren't even looking for a new species. They were collecting some other midges, and then they found these ones. Well, what do you do when you're looking for a pheromone of this particular insect? Well, you collect the volatiles. And this is a very simple setup. You just have a pump which sucks air through the whole system. You have these air purifiers, which is just simply charcoal. And this takes the volatile compounds out of the air that might be just in the lab somewhere. You have your midges flying around in here and hopefully emitting a pheromone. And then you have a dry resin in here, which will collect the organic compounds that are given off. And it doesn't need to look exactly like this. You can have various different types. But in here, we actually have sugarcane, which is what you can see. But what's hidden is this little fella, or not so little. He's actually a type of scarab beetle called an Oryctes rhinoceros. And, well, so named because of the giant horn that he has. And funnily enough, he actually managed to escape a few times in the lab and scared the bejesus out of some of the techs. But once you have collected your volatiles, all you simply do is elute that resin with a little bit of solvent, and then you have a collection of the volatiles. Then you run something called a GCEAG, or gas chromatography electroantennography. And this one takes a little bit of explaining what's going on here. But we have this diagram here. You have your injection of the collection that you just made. You put it into the GC, and then it whizzes around this coil, which can be up to 60 meters long if it was all in one line. And the idea here is that the more volatile compounds will come out quicker. So you'll actually separate all the different compounds that you collected in these collections. And half of it will come out here into a detector, and you'll get a series of peaks. And half of it will get blown over the antenna, which you can see in real life here. Here we actually took the antenna off the insect, but sometimes you can lose live insects as well. And the reason why we're blowing these compounds over here is because once there's a compound that the insect will detect, a small electric charge is produced. And we actually measure it using these electrodes. So what does that look like? Well, you have this, this graph, which is a bit confusing, but I will endeavor to explain it. The important thing is where this letter B is. And you can see there's a little bit of a peak on the 3 graphs above it. And these 3 lines above it are actually the electrical response. And the reason why you run multiple of these is because you can get a lot of noise, whether it's from a phone or another electrical device. But this one along the bottom is all the different compounds that we've collected previously. And the peak that we were most interested in is this one at B, and you can see the stars there as well. So what do you do when you think you've identified this pheromone? Well, you have to work out what it is. And you use something called a GC-MS, or gas chromatography mass spectrometry, which works in the same way as the GC-EAG in the first bit. But instead of passing it over electrodes, it puts it into the mass spec. I did wonder whether to explain what a mass spec is and how it does what it does, but I figured that the talk would take probably about twice as long. So we'll kind of slightly skip over this. But you produce a spectrum like this, where the highest number is like the total mass, and then you can actually gain structural information from the fragmentation, i.e., all the smaller masses that you can see. And ultimately, we thought it produced this compound, which sort of would fit. It's quite a volatile compound, but there was a problem. And that's these 2 things here. Now, why is this a problem? Well, this will get slightly technical, but don't worry if you don't get all of it. This is something called a stereogenic center. And when a carbon has 4 different constituents, this is an example here that's not to do with our compound, you can have a mirror image. So like this. And these mirror images can't be superimposed. So the easiest way to think of this is like a left hand, right hand. So you have your left hand, you have your right hand. There we go. But you can't superimpose them. So why is this important? Well, chemically, these 2 compounds will be almost identical. But in a biological system, it will be like you came up to shake someone's hand, and instead of using their right hand, they use their left hand. You just know something is wrong. So when these 2 insects are attempting to mate, they wouldn't accept this pheromone. So if we think of this as a right hand, and this is the left hand for this case, we had to chemically synthesize these target molecules. And this is it here. And I won't go through all of this because, well, it's quite chemistry. If you just assume it's probably the best chemistry you've ever seen, we'll get along fine. But ultimately, I synthesized these 4 different compounds, even though they're almost chemically identical. And then we compared it to the original collection. And as I hope you can see here, the starred peak is the one we're after, and it was the RS peak, or the right-left. You may think that the peak to the right corresponds to the RR peak, and it is very close, but the mass didn't quite match up. Plus, it's in both the male and the female of the species, so it's unlikely to be a sex pheromone. So the next thing is you test it. You take it out into the field. So you have a trap like this, where you have the same kind of rubber septum in the middle there, and this yellow plastic is just a sticky trap. And you see how many midges you find. So this is what happened when we first test this. And initially, this looks fine. The RS, it seems to be collecting more. But if you look on the left axis, you can see we caught on average about 0.4 midges per week, which is not a lot, and is basically could be down to the color of the trap or anything. So we went back to the starting board, had another look, had a third look, fourth look, and then we thought that maybe, just maybe, this peak has a tiny shoulder. And then if I pull up this, you can see that just maybe there is something there. And we convinced ourselves with the mass spec that there might be something there. But it's in a very small amount. So we tried it again with different ratios of the RR, or the right. And then we actually got over 240 midges per week using this. And as you can see, there was almost nothing in the other ones that we tried, even when it had the RS and the RR in sort of different ratios. There was just almost negligible. So we called that a success. But you may be asking, why even do any of this? Like, why study pheromones? Well, I'll endeavor to answer you. Well, the first and possibly most obvious is monitoring. And if it's a pest species, particularly crop pests, you can set these traps in the middle of the field. And if you can't find anything in them, then you don't have to spray your field because there's not a pest. However, if you do find something in them, then maybe it's time to use your chemical pesticides or any other techniques that you have. But it can also be used to monitor for biodiversity. So in some of these cases, you don't actually want to hurt the insect. And this is the case with this video, which is of Zangidae moths, if I got that correct, or burnet moths, to give it its more common name. And as you can see, these moths are going a bit crazy for it. But these moths are quite rare. So this is kind of like monitoring to see where these moths have survived in the UK. And the other method is another sort of crop technique where you actually try to protect the crop by mass trapping insects. And these take various different forms. So in the top left, you have sort of the more midge type traps. In the bottom left, these are sort of more beetle weevil types. The one on the right is actually for the, can't remember the name, so I'll ignore it. I'll move on. Another technique is something that I think is quite cool, which is mating disruption. So this is the moth that we had before, where we have the female on the left and the male on the right. And the female emits the pheromone, just like normal. But this time, we have pheromone dispensers out. And these are also emitting exactly the same pheromone, possibly in greater amounts than the moth is emitting. And as you could probably see here, the male moth has absolutely no chance of finding out where the female is. So in this way, you, as the name suggests, disrupt the mating. And over one generation, you can greatly reduce the pest species population. And another technique, which we've started to get into that I think is quite cool, is something called an auto-dissemination device. What's the idea here? Well, we have a friendly caterpillar, and he goes into the trap, but this time he goes out of the trap. It doesn't collect. This is more like a catch and release system. But this time, the caterpillar is infected. And he can pass that infection on to the general population. And the thing we've used to affect sort of caterpillars, or whatever we want, is something called entomopathogenic fungi. And this is essentially just fungi that infects insects. And this is kind of almost a replacement for traditional chemical pesticides. It's had moderate success, I would say. There are some problems with it. It takes longer to infect the insect for a start. So if your insect will destroy your crop in 3 days and it takes 3 days to work, it's not very effective. But it's our hope, but using this sort of combination of pheromone and EPF, we can get a longer lasting control. And now on to something a little fun. From the name of the talk, you may be wondering, are there any human pheromones? Or, i.e., is the Lynx effect real? Well, I did have a quick literature search. It's not a thing to do with my work, but I found this paper from 1991, which was published by the University of Utah, where they believe they found 2 pheromones, one corresponding to male and one corresponding to female. And this is kind of rumbled on in the background where no one has really said whether it's actually a pheromone or not. And it wasn't until these guys from the University of Western Australia came along that this was really tested in a sort of robust scientific way. And what they did is they had these 5 photos and then they doctored them using Photoshop and stuff, and sort of mathematical models. And the idea was that they made all of these 5 faces completely gender neutral. And then they subjected participants to these different pheromones, one or the other, or a control. And they were asked 3 questions: assign a gender, rate their attractiveness, and rate unfaithfulness. This one's kind of interesting because you possibly wouldn't expect it. But I think the idea was to rate the acceptability as a mate going forward. And after all this testing, what do they find? Well, there was essentially no effect, which kind of implies that the Lynx effect isn't real, which I know will come as a massive disappointment to teenage boys everywhere. All that's left for me to do is to acknowledge people that I've worked with. On the Midge project in particular, there's sort of my line manager, Professor David Hall, who did the initial assignment of the structure. Dr. Dan Bray, who did the EAG work. A collaborator in Canada, Dr. Boyd Murray. And then the other members of the chemical ecology group, Dudley Farman, Dr. Mandela Fernandez Grandon, Professor Phil Stevenson, who's actually the head of the chemical ecology department, and our newest hire, Dr. Victoria Woolley. And I'd like to thank all the different people that actually funded this work. Can't do it without them. And everyone else in the NRI. They actually made us wear these t-shirts. Wouldn't normally. And thank you for listening. And I think we'll reintroduce Connor.
Thank you very much, Steve. That was really informative. Now I'm worried that my very posh deodorant purchase is just a waste of money. Okay. So before we move on to our next presenter, I'd like to remind people that you can ask a question in the comment box to the side. If possible, please let us know where you are so we have a rough idea of whom we're talking to. Okay, so our next speaker, Dr. Sarah Arnold, is an entomologist who studies, amongst other things, the behavior of insects and how they interact with their environment. I personally have seen how excited she gets when we find a very rare bumblebee on a field trip. We didn't have a decimal meter that particular day, but my ears are still ringing from her shouts of food. Over to you, Sarah.
Thanks very much for the great introduction, Connor. So, hi everyone, I'm Sarah Arnold, and in my talk, you guys, you audience out there, are a really important part of this. So what I'd like you to do is get in the comments wherever you're watching, and you need to vote on some things for me. So firstly, I'd like you to vote for your favourite colour of flower, whether it's blue, yellow or red. And secondly, I'd like you to pick your poison, so caffeine or nicotine. Which one of those would you prefer? I'll be calling on those later because that will control the direction that this talk goes in. So, who am I? I work on pollinators primarily, and in general, I'm happiest when I'm running around in a field looking at insects, which is where we're going to go now. We're going to go to a wildflower meadow, and it looks peaceful. It looks tranquil, but actually there's fierce competition going on out there. All of these flowers, they just want one thing. They want to receive as much pollen as possible from plants of the same species as them, and they don't want that contaminated with other species out there. So that means that they need to not only win over a pollinator, but win over a loyal pollinator that keeps on coming back and sticks to their species. And going about designing a flower, if that was something that we wanted to do, in order to do that, we need to know a little bit about our pollinator. So let's introduce our first hero, Bertha the buff-tailed bumblebee. So this is a common UK bumblebee species. She's got really good colour vision, she's got fantastic sense of smell, and as insects go, she's super smart. She can learn pretty much whatever problem you present her with. And that means that she'll tend to stay quite loyal to flowers if they make it worth her while. So as a flower, if we were designing a flower for her, what do we need to think about? Well, the first thing is that she's got trichromatic colour vision. So much like us, she's got 3 types of receptor in her eye that enable her to see colour. But whereas we see blue, green and red, she sees UV, blue and green. So this means she can see the short wavelengths better than us, but the long wavelengths not so well. And that's important when you're picking a colour for your flower. She's also got quite low visual acuity compared to humans, but she's got a good memory. So imagine that she's kind of a little bit short-sighted out there, but pretty smart. There's 2 elements or 2 strategies that you could take as a flower to try and lure in that pollinator. You could exploit the innate preferences of the bee. So a naive bee straight out of the nest, what's she going to go for? What are the kind of things that appeal to her basic sensory abilities, her basic biases? But bees are smart, as we said, and if there's a new type of flower that appears in the wildflower meadow that's easy for her to find and it pays off with a suitable reward, so good pollen or good nectar, then because bees will tend to learn and compare, they'll learn to visit this flower, even if it's not perhaps the most innately attractive, they'll learn it. So effectively, they're comparing 2 different offers and going for the one that's better. So, how's it going in the comments there? What sort of colours do you want this flower to be if we're trying to make the good flower for Bertha? Okay, viewers want a blue flower. Great choice. So we're going for blue. This is a really good choice. So Bertha's got a fantastic sensitivity to blue light, and she's also got a bit of a strong blue preference when she's naive bee. So that lures her straight in. It's easy for her to pick out against the background. So if you're blue flower, you'll probably lure Bertha in, you'll get loads of visits, and you'll probably get loads of visits from loyal insects that have visited lots of flowers of that same species. You get pollinated, you set loads of seeds. Fantastic. So it's not just about colour though. There's other things that a flower can do to make themselves more attractive to Bertha. Odour is a big part of insects' attraction to flowers, and this can be particularly important when we're getting into complex environments. So if the flower is something that's harder for Bertha to see, if it's something that doesn't contrast well with a complex background, then adding a little bit of odour in there can make it much easier for Bertha to find those flowers. So odour and colour combined can be an amazing combination. But what does a flower smell of? Well, there's thousands of possible chemicals that could make up a flower odour. And the combination is important. So it's a little bit like an orchestra. Each of these different chemicals is like an individual instrument, and together they make up this fantastic symphony. But if you start changing them around, you get something that's like qualitatively different from the point of view of the receiver, from Bertha's point of view. So ratios of these different compounds can be important. Concentration can also be important. So that chemical that I've got at the top there, that's skatol. At low concentrations, that smells like, it's got a faint floral smell, it's very sweet. It's one of the components in the odour of jasmine flowers, for example. At high concentrations, it smells like poo. So concentration is important. And that brings us to our second hero, Madge the midge. Madge is a bit different from Bertha. So she's a much smaller insect. She's about 2 millimeters long. So she's got small eyes and a small brain. This means that she's much more dependent on olfaction, so perceiving the world through odour, compared with Bertha. So if we're designing a flower for Madge, what's a midge normally looking for lunch? So we want to think about what her innate preferences, what her biases might be. And I guess if you've ever been camping in Scotland, you're probably aware that midges are quite often looking for you. But there's a range of different midge species. So within this family of Ceratopogonidae, you've got the genus Culicoides, and those tend to be the itchy, bitey ones that you get around lakes in Scotland. But you've got other species. You've got the genus Forcipomia. Some of those bite us, but some of those bite other insects. So flies, for example, or true bugs. And so that means that they're using slightly different cues in order to find their host. Dasyhelea species tend not to bite at all, so they tend to be exclusively flower visitors and just eat nectar. So all of these species can visit flowers, but Forcipomia and Dasyhelea are the ones that are most likely to. And that's where it gets exciting, because those are the species that tend to visit the flowers of the cocoa tree from which we get chocolate. So if we're designing some flowers for the cocoa tree, I'd like to call Connor back and ask him to vote. Connor, if we're designing some flowers on the cocoa tree that need to be pollinated by Madge the Midge, do you think we should go for visually appealing flowers or odor appealing flowers?
Well, Sarah, as someone who is blind as a bat, as Specsavers will tell you, I really am very jealous of people with good eyesight. So let's go with visual appeal.
Okay. That doesn't work so great in practice. So Madge has got, with her tiny eyes, she can't really see the flower from her resting point even a few meters away. So unfortunately, if you're a flower of cocoa and you're relying exclusively on Madge's visual attraction, you're probably not going to set any seed, and we probably won't get any chocolate. But what about odour? Well, let's explore this. Let's introduce 3 midge species from Trinidad. So these are some ones that we caught from cocoa fields in Trinidad: a Forcipomia species, a Culicoides species, and Madge the Midge, Dasyhelea bulgmiri. They're all about 2-3 millimeters long, so they're tiny guys. This is a cocoa flower. So if you look at it, it's kind of creamy white. It's beautiful. It's an amazing, delicate flower, but we're talking about something that's about the size of a 10-pence piece. So it's not showy. It's not really using much in the way of visual attraction. And if you look at them on the branch of a tree, you could quite easily miss them, even with good eyesight, never mind Madge with her not particularly great eyesight. So we started to think about the odours of cocoa flowers and perhaps how they were using them in order to lure in a pollinator. Steve has already provided a cool introduction about how you sample odours from the environment and how you analyse them. So we did just that. We sampled the odours of a cocoa flower in the middle of a cocoa field and brought them back to the lab, where we ran those odours through a GCMS. And that gave us a graph. So this graph kind of shows those chemicals that make up the odour of a cocoa flower that Madge might be able to use in order to find those flowers. And what we learned is that these are 2 of the major chemicals that make up the odour of a cocoa flower. And these are alkenes, and they're not particularly smelly compounds compared to some of those ones that we looked at earlier. So they have a little bit of a scent, but it's very faint. But the way I describe it, if you can smell it at all, it tends to be a little bit like feet. But maybe that's telling us something about how the cocoa flower is exploiting innate biases in midges, because we know that midges tend to go for things like cues that are to do with body that they might want to land on and suck. These compounds also look a little bit like some of the compounds that you get in the cuticle of an insect. So if you're a midge that bites other insects, this might also help you zero in on that. So maybe these chemicals have tapped into a pre-existing bias that's in the mind of all of the midges, and it's going off that. Well, our first question is, do midges like these odours? So we got those 3 species of midge and we bred them up in the lab. And this graph, basically the bigger the blue bars, the more the midges are liking the odour of cocoa. So those are our 3 midge species, and all of them, to a greater or lesser extent, tended to like the smell of cocoa. It wasn't completely consistent, but basically cocoa flower odour, those compounds from the natural flower tend to work. can we synthesize that in the lab and create our own sort of super cocoa flower bouquet was our next question, because this would be a really cool thing for producing more chocolate. We could put these things out in the cocoa field, these super bouquets. It could draw in midges from far and wide. We'd get fantastic pollination and we'd all get to eat chocolate all the time, which is probably the best bit. So in this case, the dark bars, the bigger they are, the more they like that synthetic cocoa flower odor. Whereas bigger pale grey bars implies that they're going for the control, which is just a solvent. There's nothing about that should be exciting. And unfortunately, when we tested our midges on these synthetic cocoa flower odours, they just didn't really go for it. So this implies that there's a lot more to this odour bouquet than we originally thought. It's quite possible that, much as with Steve's pheromone, there are minor components that we haven't identified yet. That may be where we need to be looking next. So insects interacting with flowers and the odours involved, it's complicated. Here's another example. These are two species of flower that are closely related. The pink one's pollinated by bumblebees. The red one is pollinated by hummingbirds, which are not particularly odour orientated. And as a result, even though they're quite closely related, the hummingbird pollinated one has got a much more simplified odour, a simplified bouquet, because it's all about the visual for hummingbirds, whereas the bumblebee pollinated one has retained this more complex odour profile. Other examples from nature are things like the carrion and the corpse flowers. So these have evolved to smell like rotting flesh, so that flies and things that tend to lay their eggs on things that are rotten will fly in and try to lay their eggs here. So it's a kind of mimicry. And in the process, as they fly from flower to flower, they're providing a pollination service. Here's another example. This is the fly orchid, and this emits a sex pheromone for a wasp. So the poor male wasp flies in thinking this is some sort of super female, mates with this, discovers that that's not really working out for him, but in the process gets covered in pollen, so that when he flies off and is conned into visiting yet another fly orchid, then he's pollinating these flowers, and he's not getting anything out of this. It's just completely deceptive. Now, I asked you all earlier to pick your poison, and what we're going to do is try and drug Bertha with one of these. So, are we going to feed Bertha caffeine or nicotine? Overwhelmingly, everybody wanted caffeine. Right, so we're going to give Bertha some caffeine, and we'll see what happens. Caffeine, amazingly, when you give it to bumblebees, it doesn't affect their sensory abilities. So her seeing and her visual ability and her olfactory ability stay the same. But it seems to make her learn a little bit faster and retain memories for a little bit longer. So, why might this be interesting? Why might we care? Some research that I've been doing with some of my colleagues at NRI, working with the horticulture industry, and particularly pollination of soft fruit, has been seeing whether caffeine can be used in this. So the bees that are used to pollinate strawberries, raspberries, things like this, are usually commercial pollinators. You buy them in boxes. They have an artificial sugar solution is part of those boxes. And because they're commercial bees, we want them to stay on the crop. We don't really want them wandering off and using wildflowers, because then they're competing with wild bees and they're not offering the grower of the crop any sort of service. So if we can tinker with that food mix a little bit in order to keep the bees focused on the crop, that's a great outcome for everyone. So we tried putting some caffeine into that food and seeing whether that helps Bertha to learn the smell of the strawberry flowers better and stay on the crop. And sure enough, in some of our polytunnel trials, the bumblebees that received caffeine tended to stay on the crop more relative to distractor flowers, and ultimately out of it we got bigger and better quality fruit. So that's a really good and exciting thing that we're hoping to look into more, to see whether we can apply caffeine to give growers the edge in their pollination. So getting pollinated is not that simple. You can't just be a flower and hope for the best. There's competition out there. There's only so many pollinators, and if they get distracted, they're dumping all sorts of other species pollen onto your stigma and clogging it up. So you don't want that. So there's a lot of benefit if you can win a pollinator and get a loyal pollinator. it might be useful to be distinctive. So if you're easy to recognize in a noisy, busy environment, whether you're doing that with color cues or odor cues, a combination of both. Or you could exploit the innate preferences of your pollinator. So appeal to what they're naturally going for when they've had no prior experience. So there's all different things, and I hope you enjoyed this. I'm just going to acknowledge all of the people who made all our work possible. And thank you all for being a fantastic audience, and thanks for all your voting. That's made it extra fun. Back to Connor.
Wow, thanks, Sarah. I'll be honest, I was looking forward to some chocolates, so slightly disappointed. Okay, so we're going to, before we jump into the questions, I'd like to remind people there's still time to ask a question in the comment section. Please, if you can, let us know where you're from. So while we're waiting for some questions to come through, Sarah, can I ask a question? And
sure.
Steve.
Hey. I'd be honored to answer some questions.
So we see in the media increasing articles and stories of that certain insects are maybe smarter than we thought, or having traits and gifts that we might not have expected. Do you think this is because up until relatively recently, we really haven't been given, haven't had a full appreciation of just how amazing insects are?
Yeah, I think there's a lot that even those of us who work on insects are yet to learn. And whereas people are probably quite familiar with something like a bumblebee that's large and distinctive and beautiful, and people understand somewhat about what they do, it's perhaps a little bit harder for people to see the appeal of something like a wasp or a fruit fly. But actually, they've got equally elaborate nervous systems. They've got amazing sensory capabilities, and some of them are fantastically smart. I mean, a wasp can learn probably pretty much everything that a bee can learn. It's just that people like bees a lot more than they like wasps. But any insect, I think, once you start to study it and realize how elaborate and fantastic it is, you can get excited about it.
Okay, brilliant. And we have our first question in. Dave Marshall wants to know, are there any examples of predators exploiting pheromones to track down their producer?
Yes, multiple actually. And I can't think of any of them at the moment, but yes, it's quite sort of a reasonably well-known phenomenon where my colleague, Professor Stevenson, actually released a paper called something like Insects in the Arms Race. And you kind of have this trade-off about how visible you are in olfactometry terms. So in certain circumstances, if you're super visible, i.e. you're producing all of these different compounds, you can attract the wrong thing. So yeah, it can be a bit of a trade-off going on.
Brilliant. Thanks, Steve. We have another question from Dawn Holmes. Dawn wants to know, can pheromones or scent be used to deter insects from an area, to protect crops, or to form a fence to keep bees in?
Ooh, there is some research in this sort of area. So there's a system called push-pull that involves the idea of something repellent and something attractive. So perhaps something amongst the crop that repels insects by emitting a volatile that they don't like, and then attracting them into something around where they can't cause any damage. The difficulty with anything that's repellent is that if there's enough reason for the insect to come and use that crop anyway, then they could potentially learn or adapt and start ignoring that odor. And if you expose insects, I think, to an odor for too long, then sometimes they just stop responding. The same as if you're sitting in a house with a bad smell. After a while, you just kind of don't notice it anymore. So you have to be careful. So yes, you do have to be careful about deploying repellent odors. Steve, have you got anything else that you would comment on there?
Yeah, it's kind of echoing what you were saying. Repellents are useful, but they tend to have a limited lifetime. After a while, insects will stop paying attention to it, because it was actually the same with alarm pheromones, where if you apply it, then they act like there's a predator. But if you just have it all the time, they stop reacting to it. So there is use, but it can be a bit limited.
So I guess, are we saying that the insects are clever enough to twig that there isn't an actual apparent threat in their immediate vicinity?
Yeah, how intelligent certain insects are is a matter of some debate that is beyond my capacity to answer, I'm afraid. But they certainly, after a while, do learn if something isn't benefiting them.
Okay, we have another question in from Becky Blunden, and Becky wants to know, how do you make sure this type of research doesn't damage the natural world, and how it's worked for many thousands of years?
I think that could apply to both me and Sarah, but I will answer it sort of from a more agricultural perspective, where one of our goals is to reduce the uses of chemical pesticides, which does have this quite damaging effect if overused. There's this thing called integrated pest management, where it has a pyramid, and at the top is the chemical. So essentially that is the last resort, and everything else is sort of leading up to that to try and prevent it. I think I will let Sarah answer about sort of introducing pollinators into what would previously be wild pollinators, I would guess.
Yeah, I think the thing that we really need to remember is that modern agricultural systems have already incredibly damaged the natural world and are very much removed from all of the ecosystems that evolved naturally prior to human inhabitation. So before we started messing with the environment, you wouldn't get a plantation of just cocoa, you wouldn't get 6 acres of strawberries in polytunnels. So we're starting from an artificial system at the start. And things like crop pollination, quite often natural pollinators just won't cut it. And that's partly because we as humans have just destroyed all of their habitats. So local populations have dropped down in a lot of agricultural and horticultural regions entirely. And partly because if you're just going to plant crops at that density, the numbers of pollinators that you'd need to do that without introducing anything would be enormous and very difficult to sustain, because crop flowers, for example, might only last a few weeks, and what are the pollinators going to eat the rest of the time? So we're already starting with something really artificial. So it's necessary perhaps to bring in something like managed pollinators, whether that's the honeybees that pollinate almonds in California, or whether that's bumblebees on strawberries. And certainly the commercial bumblebees are quite an artificial system. They're out for maybe 6 weeks. And then at the end of that, they tend to be taken away from the field. So what we really want is for any interventions to minimize their interaction with things beyond those polytunnels. So it's artificial, but if we manage it in the right way, it can be minimally damaging. And that's what we're aiming for, is kind of tweaking the environment a little bit to, as Steve says, reduce uses of chemicals and reduce how our actions kind of impact other areas that are perhaps less touched by human interventions.
Oh, that's brilliant. I guess it just reinforces that sometimes it's actually quite difficult to distinguish between what is natural and what is organic or living or buzzing around, but not necessarily a part of a natural system. Okay, we have a question for someone else, for Sarah, called someone's from Hans Dampf. Sarah asked Hans, how do you feed caffeine to the bees in the field?
Okay, so the commercial bumblebees, they come in a box and there's a reservoir of sugar solution underneath that. So that's so that if the bees are deployed on something like tomato, which doesn't produce nectar, the bees can still feed. And that is an artificial sugar solution. So there's lots of things that potentially one could do with that sugar solution in order to boost the bees' performance, keep them going for longer, improve their health, stop them getting diseases. So that reservoir has got a whole load of potential in terms of keeping those managed bees happier and healthier.
Brilliant. We have another question from Stephanie Pike. And Stephanie, do you think there is any grounds in the dating sites that send you a scent sample of a potential partner? Oh, I'm not going to touch that one.
Well, Actually, assessing whether this is a real scent or something they just sprayed in there would be quite difficult. But I think it's an interesting idea anyway. Maybe we can set up a human entrainment system where we collect the volatiles given off by potential dates. Make millions.
There's an app in that. Okay. All right. A question, I think, for Sarah from Becky. And Becky wants to know, how does nicotine affect pollinators, or other choice that we didn't go for because we far too much?
It's a good question. Unfortunately, I can't take credit for work that went into this, but so a research team at Queen Mary did look at this, and they found that nicotine does also make them perform a little bit better, so they remember stuff a bit better, they get more motivated. Unfortunately, nicotine is also horribly toxic, so it will probably have quite bad impacts on the bees' health, either in the short or the long term. Whereas in our experiments with caffeine, so far we found no evidence that feeding bees low-level caffeine does anything bad to them at all.
Right. Okay. Thanks, Sarah. A question from another Sarah. Again, I think it's more directed to you. Too many Sarahs. Sarah wants to know: can insect pheromones be used in a positive, i.e., behavior-promoting way, rather than a negative, behavior-preventative way, such as increasing pollination? And I think we've covered this already. But is there anything you want to add on the positive or negative reinforcement of pheromones?
I suppose one thing that's quite cool and is being looked at in some areas of research, not specifically pollination, but in pest management, is the idea that when an aphid is attacked, then it tends to send out pheromones, or when an aphid's feeding, it can send out pheromones, and those can also be picked up by parasitoid wasps that fly in and lay their eggs inside the aphid. So there is some work that's going on into, can we recreate that and make artificial systems that lure in those parasitoid wasps before the aphids get to a high pest damaging population? In terms of pollination, it's a bit more tricky because most pollinators are not using insect pheromones in the process of pollination. The exception is those sexually deceptive ones, but most of the time when an insect's got its mind on pollination, it's not really responding to things like other insects' sex pheromones.
Okay, we're nearly run out of time, so I think we have time for one last question, and we have a question, I think, from our colleague, I'm guessing, Lionel, unless it's a different Lionel. Says, thanks, Steve and Sarah. Yes, thank you very much, Steve and Sarah. Sarah, did you say that a better pollination system can increase the size of fruits? If yes, how does it work?
So strawberry is made up of like lots of individual little seeds on the outside of the drupe. And if some of those are not properly pollinated when they're at the ovum stage, then the flesh around that doesn't develop properly. So you end up with like a little scrumpled up misshapen strawberry that nobody really wants to buy. If the whole of the strawberry flower has been evenly and gently and effectively pollinated, then it kind of grows out nice and evenly and becomes big and even and beautiful. So it's effectively about getting pollen spread nicely over the surface without the bee going kind of too excited and chewing at it or anything like that.
Okay, well, thank you, Sarah. Now I'm hungry for a cup of coffee, a bar of chocolates, and some strawberries. Okay, I think that's just about all the time we have for this evening.
Science.
I would like to remind people of the Twitter hashtag Pint of Science 20, Pint 20. And I would like to thank our fantastic questioners. And I would like to, of course, thank our amazing presenters. Thank you, Sarah, and thank you, Steve. Okay, folks, whether you're watching live or you catch up, I hope this has been interesting and informative. And I hope that you join us for the next session in about half an hour entitled Food for Thought, which is appropriate because I'm certainly getting hungry. Thank you all. Have a great evening.
My name is Adam. I will be your quiz master for this evening. I certainly hope you're going to enjoy yourself. Here we go.